Hi, everyone. My name's Kristen Rosamond, and I'm a member of the AOS Student Affairs Committee webinar series team. Thank you so much for joining us today for the Manakin Mini Symposium. We're all really excited about it. And just as a reminder, the webinar is being recorded and it'll be posted and available online afterwards if you want to view it again. So I'm excited to introduce Dr. Robert Driver, and he recently graduated with his PhD from East Carolina University. He studies the molecular evolution of gene families that are involved in the sensory perception of birds. So I'm going to go ahead and pass it over to Robert now, and he's going to get us started with the event and introduce our panelists.
Okay, yes, thank you, Kristen. And I'm just going to share this very quick introductory PowerPoint before giving time to the speakers. So I just wanted to talk about this Manakin Mini Symposium that we're having. It's featuring the work of members of the Manakin Genomics RCN, which is a research coordination network through NSF. And I just wanted to tell you guys a little bit about the RCN if you are not familiar with it. This is a snapshot, not full picture, of some people in the RCN and the species of manakins that they study and that we study. And our RCN is diverse across studying all sorts of manakin species, as you can see here across the phylogeny. We're diverse in institutions with members from research-oriented institutions, teaching institutions, government facilities, as well as diverse in our location with scientists in the Research Coordination Network from North America, also South America and Central America, and Europe across the world. So we're happy to have all these researchers studying manakins together collaborating. We're also diverse in the fields that we study. So there's ecologists in our network, animal behavioralists, as well as evolutionary biologists and genomicists. So the goal of the RCN is to take all of our focuses and combine them and collaborate to answer bigger questions. So to do this, we've had activities in the past. So there have been used to be annual Manakin RCN meetings before the pandemic. We've also had symposia at major conferences. So at NAOC, there was a symposium, and at SICB, there was also one. And this is our two most recent in-person meetings were in Brazil, as well as Panama. This is a picture from the Panama meeting. And actually in this picture, just interestingly, Lia is next to Kira, and I'm here behind Camilo. So it's like the presenters for today. Yeah, anyway, so we also have some resources here that you can check out if you're interested. Members of the RCN have sequenced 6 different manakin genomes, which are available on GenBank, and they're high quality and they can be used to answer all sorts of questions. Associated with the SICB Symposium, there was a volume in integrative and comparative biology in October 2021, where you have 10 Manakin papers to check out there. We also have a website, manakinsrcn.org, where you can see nice pictures of manakins and learn about our research. And also, if you search Manakin Genomics RCN on YouTube, you will see many pre-recorded talks from those meetings and symposia that I showed in the last slide, so you can catch up on the work that has been done by our group. So that's it. I'm going to stop sharing and we'll start with Camilo. So I forgot about Manicus candii, Kevin says. So, okay, 7 genomes. Sorry about that. And 2 in Manicus. Now, I did have Manicus up there. So, okay, so we'll start with Camilo. Camilo got his undergraduate degree at Universidad Nacional de Colombia in Bogota, Colombia. And Camilo has been researching manakins for a long time, so maybe 15 years or longer. So he, as an undergraduate, he researched ecology and behavior of Neopelma chrysocephalum. And then after he graduated, He did a ton of field work, different technician jobs with the University of Wyoming in Panama, as well as with the Smithsonian in Ecuador, before starting his PhD at Virginia Tech in Ignacio Moore's lab, where he has had amazing funding for his PhD, getting a Fulbright, which supported him for the first 4 years, I believe, and then an interfaces, of global change fellow, which he is supported by currently. So I will let you tell him here about the Manakin work that he has been doing for his PhD.
Thank you so much for that kind introduction, Robert. Yeah, hey, hello everyone. Today I'm gonna tell you a little bit about like what is my research with manakins. You guys can see my presentation, right?
Yep, looks good.
Okay, and what about the laser point? Yes, it's working,
Yes.
the laser. Okay. Sorry, I just was double checking. So why manakins? So part of the natural history of manakins is that they have like leaky behavior. Manakins aggregate in areas called the legs to perform courtship displays that attract females. Females evaluate males, male performance to choose what male they're gonna mate with it, but they don't form like long-lasting pairs. So manakins are polygynous, and female mate choice drives a strong sexual selection in all manakin family that has produced a diversity of traits and display behaviors. Since manakins do not perform like do not form pairs, females take care of all parental care alone, and that allows males to remain in the legs, to perform displays, to keep attracting more females, or interact with other males to define hierarchical positions, to define like in other species social conditions. I will like talk about it in a bit about that more. So with all the manakin family, there is like a complex and diversity of social behaviors. Like despite besides all like the diversity of like display behaviors, social behavior is interesting in manakins too, because we have manakins that go like from like complete like alone like displays, like the red cat mannakin of the golden color mannakin, where like they go and display alone to attract females. But in other species can like form these social coalitions between two different males, and that's the example of the Xeroxipia males and the Pipra males. So the focus of this part of my research is this diversity in social behavior, and specifically I want to like explore the relationship between cooperative behavior and aggression. So as I mentioned before, within the manakin species, two genera have been reported to show a type of cooperative behavior where unrelated males form social coalitions with well-defined hierarchical structures. So in some like species, there is also like they also like do like join or like interact with other males, but something that is unique between these two genera is that they have like a well-defined hierarchies that like are relevant for the social interactions. For example, in the wartail manakin, they go in like two different stages. We have like floaters, and floaters can be usually like young males that don't have like definitive plumage, or some of them have like the definitive plumage. And then we have territorial males that are the males that acquire a territory and like can mate with other females. So for the wartail manakin, having a territory is like super important, because that's how like they can like mate and have like reproductive success. Something really important for manakins is also how like males interact with each other, and they have like this with this coalition that form like a complex social network where like different males have like different sets of connections, and these connections determine the reproductive success of a male. So, but they also determine like if like a floater male, a male with no territory can acquire a territory, if depending on the number of connections that they have. And in both type of manakins, males spend long periods of time in this predefinitive plumage or this predefinitive social state that might help to reduce aggression from the dominant males or the males that have a territory. So with that in mind, like my question is, what is the role of aggression in the social behavior of manakins? And like some reports, like some reports have say that in most species, aggression is not commonly observed. Or if it happens, it is usually controlled, like in some species. Still, we know like this like aggressive agonistic interactions are happening at the legs, because that's probably how males define hierarchical positions, or how like they like make other males to respect their territories. So to answer the question of the role of aggression, I designed a set of experiments that I will explain shortly. But first, it's probably important to understand that aggression probably has a cost for social behavior. Especially that's the case for the species of males that form these social coalitions. So aggressions could affect how male-male coalitions and cooperative behavior is happening in the Wertel Manakin, because if they're like too aggressive, they might not be able to form like social coalitions with other males. And if they're not probably aggressive enough, it's like a little like they can like get out, like kicked out of their own territories. So for that, my hypothesis is that male species that form social coalitions are less aggressive during territorial challenges than species with no social coalitions. And basically, to answer like this question and hypothesis, what I did, I compared two species of manakins, which I will explain now. So my first species is the red cat manakin. Males display to females alone, and they do not form social coalitions. I conduct like my experiments at the Smithsonian Tropical Research Institute at Panama, and they're like we have localized some legs there, and like I knew where to go and where like to conduct my experiments with this manakin. And like something interesting about this manakin is like the type of leg that they have is called a condensed leg, and that means that males are like pretty close to each other, and they are within like visual contact. And it's interesting because you see when you are like doing like behavioral observation at this manakin, usually you don't see males attacking each other, despite like they are like very close to each other, and aggression is like difficult to observe. My other species is the Wertel Manakin. This is a species that form social coalitions and have like this pretty interesting social network. I conduct this study at the Tiputinity Biodiversity Station in the Amazon area of Ecuador. And as I mentioned before and explained a little bit, for like this species, social coalitions are super relevant because they are what determine their reproductive success. So basically, to like answer these questions, we perform simulated territorial intrusions. These are like pretty classic experiments in behavioral ecology, where we just put a taxidermied mound in the area of the male. And from here, we took different like variables. We got like video recordings, and we measure first direct attacks, how many times like a male attacked directly the taxidermied mound. And then the second variable that I'm taking from here is called an aggression score. And aggression score is like a different conjunction of different variables: exploratory jumps, territory calls, latency to approach, time spent with the mount, and closest approach. So basically what I did, I just took one of like these taxidermy mounts to the territories, put them in their territories, and observe how aggressive they will behave towards the taxidermy mount. And I have a couple of videos here that hopefully they're gonna work. Let me see. So this one is for the water mannequin. So here I have the taxidermy mount here. You can see that it's very static and it's not moving. And this is the territorial male. And you can see like the male is doing some like exploratory like flights close to the taxidermy mount. And there you see an attack. They hit the mount. It hit the mount. So all like this behavior like happening here. And you can see how like actually just like stand over the mountain and start like picking it. So that is pretty aggressive. And another one for this is the red cat mannequin in Panama. And this is a taxidermy mount. You can see it's very static. And the male and the territorial male is like around here. He seems also like very frustrated with the taxidermy mount at the moment. And he's attacking it. That kind of like another attack because he flew like by and like pecked it before it landed in the other perch. So yeah, that's basically how I record like the behavior during the simulated territorial intrusions. And now as the results, like very like short results here, there is a trend of the red cat mannequin showing more aggression compared to the water mannequin. Here in the y-axis we have the number of attacks, and in the y-axis we have the red cat mannequin that display alone, and we have the water mannequin that have the social coalitions. And we can see here in the number of attacks that in average, the water mannequin have like more attacks, but like the difference was not significant. And we compare also like the aggression score, we see that there was like a trend, like very close, almost significant, of the red cat mannequin being like more aggressive compared to the water mannequin. And this is the aggression score, just to remind you that this is like different variables that the mannequins, the different behavior that I record during the territorial intrusion. So as a main findings of like this short experiment, there is a trend of red cat mannequins showing higher aggression than the water mannequin. But still we observe that the water mannequin have show some levels of aggression. And we think that the cost of losing a territory might be higher than losing a new social partner. That means that if like an aggressive male comes to like a water mannequin, and he tries probably to determine if this is gonna be a cooperative behavior or like a social coalition, and it doesn't happen, but it's just like a male, but like being aggressive, trying to take over the territory, the water mannequin is gonna respond anyway with aggressive behavior. But this is also one part of my research, so I'm hoping to like conduct that more depth of like my dissertation. So the next steps, like during these experiments, I took some blood samples, and in my lab we studied testosterone. So we hope to compare how also if testosterone is higher, is any high during like these simulated territorial intrusions, and compare between the species. And we also have like some experiments with female mounts that I conducted like last year, and also have like the blood samples to see if like maybe testosterone is increasing during either like the territorial intrusions or during the interactions with the females. And just like as mentioned before, we know that testosterone is relevant for social behavior. Like a previous research conducted in our lab for Ben Bernasco, we know that males with higher level of testosterone here have lower number of cooperative displays. So we know that testosterone is going to be relevant for like these interactions, and we want to measure how relevant are going to be. And with that, I would like to acknowledge all the people that helped me with the project and my funding sponsors, and I'm open to any questions. Thank you so much.
All right, thanks a lot, Camilo. Maybe we'll have one verbal question quickly, and then anybody else can also type the questions in the chat as well, and Camilo can address them as we go along.
Yeah, you can contact me anytime at my email too, or my Twitter account.
Cool. So feel free to type. Maybe people are typing now, but we will move to Lia, our second presenter, and Dr. Lia Kajiki got started with her BS in biological sciences from São Paulo University in Brazil. She then moved far for her master's degree in Amapá in up north in Brazil to get her master's in tropical biodiversity, and then moved down to in the middle of those two locations for her PhD in ecology at the University of Brasilia. And Lia's been great at getting funding. She has she got an early career grant from National Geographic Society, which helped to fund her PhD. She has multiple publications in all sorts of ornithology journals, emu, austral ornithology, ornithology research, Caatinga, as well as other tropical ornithology journals. And also, I should mention, Lia and Camilo were very helpful with the RCN translation of the newsletters that we used to have in the RCN. Maybe we'll bring that back. We'll see. But I'm going to let Lia tell you guys about her Manakin research.
Hi, everyone. Can you hear me? Is it okay?
Yeah, it's good.
Thank you, Robert. Thank you, Robert. I'm gonna share my screen then. Thank you very much for the invitation, of course, and for all of you who are today with us here to talk about manakins. So my name is Lia. Thank you very much for the nice introduction, Robert. And so the work that I'm gonna show for you today, it's part of my PhD thesis, which I concluded in the University of Brasilia in Brazil. And it's about the helmet manakin. And hopefully it will be soon published and available for all of you to check the details if we don't have enough time to talk and discuss about it. So in Portuguese, the title is Territorialidade Individual em Espécies de Leque, o caso do soldadinho. And let's move to the next slides. So mating systems have been largely investigated under an ecological-based framework, and which is defined by the capacity of one sex to monopolize territories, resources, and mates. And in monogamy or resource defense polygyny, for example, males exclude others from resource-rich sites and consequently acquire higher reproductive success. However, when resources are abundant and cannot be monopolized, then males can aggregate to attract mates and form leks, like happens with the manakins, with most of the manakins. And this is, as you may know, is the swallowtail manakin, Chiroshia caudata, and it's a very complex display, example of a complex display. So although most lacking species fulfill the criteria of a classical lek, there is a lot of variation in the level of aggregation, of male aggregation. So when males are loosely aggregated, the term exploded lek is used. And in this type of lek, males display areas sometimes contain resources, while in non-lacking systems, males monopolize resources in their territories. So it's important to say as well that in lek systems, the resources are neither essential to females nor they influence mate choice. Or in other words, so the male itself is the main focus of the attraction of females, for the attraction of females. But classifying mating systems in nature in discrete categories may be very challenging. So for example, the little bustard, Tetrax tetrax, mating system has been interpreted as both true exploded lek as well as a non-lacking system. So these and other examples in birds, ungulates, and insects, they suggest that maybe a continuum between categories may better represent the variations in nature. So the lek system is widespread among manakins, which we all know is a family of birds of beautiful and striking ornaments and complex courtship displays. And the elaborate displays and ornaments, they are thought to increase with the intensity of sexual selection. And the other way around may be also true, in which the relaxation of sexual selection could result in the loss of elaborate traits, such as may have happened with the green manakin, Xenopipolo chlora. So a few species in the family do not exhibit typical lacking patterns, and this is the case of the Araripe manakin, Tilapia buchermani, in which females, researchers found out that females nest within male territories, but the territoriality and offspring paternity are not associated. So despite their potential to shed light on mating systems and sexual selection, these atypical manakins have been less investigated. And the helmeted manakin, Antilophia galeata, despite its striking sexual dimorphisms, also a pattern among the manakins, has long been considered as having deviated from the lacking pattern and evolved towards social monogamy, based on historical observations of pair bonding and individual territories. So these studies proposed that this deviation from a typical lacking system could have happened because of selection associated with an evolutionary preference for the gallery forests in the tropical savanna cerrado biome. And I would like to take some moments to tell more about this interesting environment. So gallery forests, they line the watercourses within the open savanna-like vegetation matrix, like this photo. And they represent only 5% of the Cerrado biome and are more restricted than typical equatorial forests occupied by most manakins. So important resources may have a patchy distribution and potential limited availability. So this unique spatial configuration could shape competitive interactions and breeding behavior of the species. And the two antelope species, they are very similar, although they have this obvious color difference. But however, while the entire population of the Araripe manakin occupies a teeny area of around 30 kilometers square, the helmeted manakin is widely distributed across nearly 2 million kilometers square of Cerrado biome. And so we think that the population dynamics should be different among the two species, affecting potentially territorial size, breeding behavior, and possibly the mating system as well. So our aims in this study were to investigate territoriality and breeding behavior in the helmeted manakin. So first, we hypothesized that males in the Antilophia genus would behave similarly. and defend individual territories with nesting sites, as it was found with the Araripe Manakin. Then, if Antilophia, if the Hamilton Manakin exhibited a resource-based territoriality, we predicted that males would be more aggressive towards the center of its territory, and nest sites would be found more frequently inside territories than outside. And we expected that if the species was monogamous, both males and females would perform parental care. But if female uniparental care was found, we predicted that males would be intolerant of conspecific intruders within their territories, if this was the case of resource defense polygyny. And alternatively, they would be more tolerant of conspecific intruders in a lacking system. So the study was conducted in a protected area in Cerrado biome in Brasilia, the capital of Brazil. We sampled for 3 years the humid gallery forests, such as the photo on the left. And the area covered, total area covered was around 1 kilometer square, and it's surrounded by savanna vegetation, as the photo on the right. We randomly searched for nests that were monitored with camera traps. We captured and banded Hamilton manakins and monitored a total of 8 banded male adults to study their territories. I won't go through much detail on how we calculated territory size and area, but I'll be happy to answer any questions about it in the Q&A session. So basically, we estimated the preliminary territories with MCPs to have an idea of the size of the potential territories. Then we conducted STIs and used the kernel density estimation technique to estimate the actual territory size. So we consider that territories, they are defended exclusive areas where typically breeding activities occur. And the experiments were conducted as represented in this drawing on the bottom right. So a mounted taxidermy decoy was exhibited with a playback protocol of 10 minutes in each of these 3 categories of distance from the center, what we thought was the center of the territory. So the preliminary estimates of territory with the MCP revealed to be conservative estimates when compared to the actual territory. When we compared the territory size that we found for Hamilton Manakin compared to the Araripe Manakin's territory, we found that average size of Hamilton Manakin territory was 7 times larger than Araripe Manakin estimates. We also found an average home range size of nearly 6 hectares for the Hamilton Manakin that encompassed the smaller territories, as you can see in this image on the right, representing one of the male's territories and home range. So regarding the aggressiveness comparing the distances, so males' probability to respond to the experiments conducted closer to the central area, they were greater than at the border of the territory. So experiments at the center positively affected the probability of an agonistic response, suggesting that the mating system could have a resource-based territoriality. And regarding the STIs itself, so we conducted 55 territorial defense experiments with the 8 males, and in nearly half of the total acute responses, males responded with this behavior in this video, in which it seems to be trying to attack the decoy. But curiously, the male never physically attacked the decoy, as we saw in Camilo's previous presentation. So we classified responses as the first approach of individuals to the decoy, and we called acute responses when the males started to approach even closer, around 1 to 2 meters from the decoy. So previous STIs conducted with true lacking manakins found comparable results, and these studies, males physically attacked the taxidermy mounts. suggesting that some level of aggression may be found among the cooperative lacking manakins. But well-established dominance hierarchy, and the Hamilton Manakin seems to display solitarily, and at the time of this work, we didn't know exactly if there was a dominance hierarchy. So the function causes an intensity of the male aggression in manakins they made lacking configuration. So although the individual territoriality we found in this species may signal it's defending important resources for females, suggesting a non-promiscuous mating system, we speculated that these territories may function as solitary display arenas instead. So regarding the nesting data, so breeding data suggests that only females perform parental care based on the monitoring of 33 nests, active nests in nearly 15 hours of video recording. And well, classic theory proposes that the parental care and mating systems are closely associated. So females performing all parental care would indicate that males have an opportunity to be polygynous, similar to what happens to the rest of most of the members of the Pipridae family. And in polygamous systems, it is expected that females will nest on sites independent of males' locations. And for lacking birds, even away from display grounds that could potentially attract predators. We found that around 20% of all active nests were located within males' territories. And although none had more than two or more simultaneous active nests, nesting seemed clustered in some areas. And we sometimes observed females nesting in the same area across 2 breeding seasons. So territorial behavior may vary as a function of many variables related to the site, to the habitat, like quality, intruder pressure, costs of defending the territory, which could affect territory sizes, shapes, and placement. And we think that this curious pattern of nesting and territoriality in the Hamilton Manakin could be a result of the habitat limitation established by the size and the shape of the habitat of the gallery forests. And regarding the display, according to our observations, males' displays of the Hamilton Manakin are basically the advertising song they perform. You can't hear, can you, Robert?
No, sorry, you might have to try an impression here, possibly.
Okay, I brought, instead of that, otherwise you would have, it would damage your ears. I brought can you hear the song? Let's see.
Everyone turn up your volumes here.
Yeah, if you don't know how a mannequin sound yet. Yes?
It barely came through once, a little bit.
Anyway, so it has a very characteristic rollicking song, and we think that this is the main way that it performs the display, by advertising it and doing around its territory, and also very simple postures, as you could see here in the video, like the horizontal and upward posture. And these postures, actually, they were already described for a few species, like the orange crescent manakin, Heterocercus orange vertex, and the simple display is also described for the black manakin, Xenopipotronitens, which share some similarities in habitat, especially with the Hamilton Manakin. So to wrap up, although we found that the Hamilton Manakin holds individual territories, only females perform parental care duties and males display in solitary arenas. So increased aggression towards the center of the territory and the nesting behavior, they may indicate that Hamilton Manakin has maybe a mating system within this lacking resource defense continuum. But of course, I need to finish analyzing the paternity genetic data to confirm the mating system. to see if it's polygynous as the other members of the family. And I would like to thank the AOS for the opportunity to share my research here, to the Manakin RCN, of course, for bringing us together and being an excellent model of international research collaboration. Once again, thanks, Robert, for leading the organization of the seminar, and to all funding agencies that support this study, and of course, my eternal gratitude to the co-authors of this work and to all volunteers who helped me to collect data. Thank you.
All right, great. Thanks so much, Leah. Looks like there's already some questions coming in, typing wise. If anybody does want to ask one, we probably have time for one. That I'm sure we were all blown away by that video of the nest coming down from the top. Really amazing, right above the river. So, unless anyone wants to jump in here, we will hear from Kira next. Kira, yep. So, our third and final speaker is Kira Long, and Kira got her BS from University of California, Davis, where she, actually, I didn't know this, where she researched sage grouse courtship. So that sets her up nicely to then go to another lekking system here. And after she graduated, she did more sage grouse work and also some field work on wood ducks. And Kira, like Leah and like Camilo, has just so much field work experience. All three of them have, I'm in awe of their field work. Kira's had a lot of field work for her PhD, as well as she's a PhD candidate finishing up at the University of Illinois, Champaign. And I will let her tell you all about the hybrid zone of Manakins in Panama.
All right, thank you very much, Robert. Can everyone see my screen as the habitual question? Good,
Yep,
all right.
I can continue to answer.
Yeah, thank you very much. So now we're going to talk about the Manakins hybrid zone. I'm going to go into some population genomics and all of that. So this talk, I have crammed an overview of my whole dissertation in here. So if anyone's got any questions on more in-depth methods, I'm very, very happy to answer them at the end of the talk or via email. So here I also have a list of a bunch of my collaborators, but there's many, many more people who are on my acknowledgement slide at the end who helped with all of this research. So hybridization is an important evolutionary process, and hybrid zones are dynamic and can change over time. So in nature, we can see both rapidly moving hybrid zones and stable hybrid zones in a wide variety of taxa, and characterizing the movement or lack thereof in these hybrid zones can shed light on how species evolve over time and how species limits are maintained. And so today we are focusing on manakins. So we have this lovely Manakin hybrid zone between the white-collared manakin and the golden-collared manakin in Western Panama. And these are our culprits right here. So previous work on this hybrid zone has noted this asymmetrical integration of sexually selected traits, namely plumage coloration traits. So notice that our hybrid down here, this hybrid boyo, has this yellow collar up here and yellow belly, which you can see the yellow belly in the white-collared manakin and the yellow collar in the golden-collared manakin are both kind of combined here in this hybrid. So females in the system appear to prefer males with these yellow collars over males with white collars. So this has contributed to this introgressing yellow plumage trait. There's also evidence of this olive belly color down here on golden collared manakins, also introgressing over time. So there is evidence of additional evidence of male aggression possibly being associated with lek placement and other things. So talking about leks, you've all heard a lot about leks today. So this is a lek mating system. And so we know here that all the females get all the mating systems, it's highly competitive with strong sexual selection on male traits. So this locking system might make this hybrid zone particularly interesting because there could be this balance of sexual selection for a trait a hybrid has against selection against hybrids. So here we have a map of this hybrid zone in Bocas del Toro, Panama. We have our white-collared range up here going into Costa Rica. On the left, we have our hybrids here in the middle and our golden-collared manakins here on the right. Here are these dots and numbers represent my sampling sites, which are actually replicating some of the original sampling sites from the 90s. So previous research on this on this hybrid zone in both genetics and phenotypes found the genetic center to be here. So population 9 had these most admixed individuals in this hybrid zone. And then we had this phenotypic transition here along the river where birds on one side are yellow, but birds on the other side are white. And so these two different transitions between genetic and phenotypic is insinuating that this is introgression. So we expect that if there's strong sexual selection in this male trait, that it'll continue progressing along this hybrid zone. If it's beneficial, it should continue to spread in populations. However, without another time point, we don't know if this yellow plumage trait is moving or not. And so that's where the study comes in. So I'm assessing the temporal dynamics of this hybrid zone. So I am attempting to determine if this hybrid zone, given female preference, has moved or remained stable over time. I genotyped contemporary and historic samples, so about 500 individuals were sequenced from genetic samples from 30 years ago, procured from the Smithsonian in Washington, DC, as well as I collected samples in the field from 2017 to 2020. I also measured male plumage traits that may be under sexual selection or approximate to sexual selection in similar traits that was measured in the 90s. So we compared both phenotypic and genetic. So what did we find? So we found that admixture here, so this is an admixture plot showing genetic clustering of individuals across this hybrid zone. So on the top, we have the admixture of the contemporary birds, and on the bottom, we have the historic. Our golden collared birds are over here on the right, so I'll use this cartoon to denote the golden collars throughout the talk, and the white collars are here on the left, associated with population 2 and 10. You'll notice here, population 9.5 I added, so there's no historic population for this, just to get a better look at this. And so what you might notice at this plot, looking at these two colors for these genetic clusters, is they're eerily similar. So this pattern is, the sample sizes are slightly different, which is why this is a little smaller, so it's a little less, fine-tuned, but the pattern is eerily similar between these two time points. Overall, the admixture, the genetic admixture is extremely similar between these two time points in this hybrid zone. So looking at this a little more closely, I calculated a genetic hybrid index, which tells you the proportion of parental alleles in hybrid genomes. So on the left, on the x-axis, you're going to see these populations again, 2 through 10. And on the y-axis, hybrid index is calculated from 0 to 1. So 0 in this case means candy eye-like, and 1 means vitellinus-like. Blue is our historic, again, red is contemporary. So very similar to what we see in the admixture plot, these populations all look really eerily similar. You can see how there's a lot of variations, population 9 compared to all the other populations. And when we look at a Kleins, this is a geographic Klein calculated for the hybrid index, and we overlay this to see if these Kleins overlap or not, and if they're moving or not. And again, they overlap eerily similar. These population averages are almost indistinguishable between these two time points. So overall, doesn't seem like the hybrid index or the overall genome has moved very much in this population at all. So overall, hybrid zone seems stable. But what about those phenotypic Kleins that I mentioned for sexually selected traits? So we measured a number of these sexually selected traits, such as these birds have beards, or the bearded manakins that they put up during their displays. Their epilep widths, or how far down this coloration for their plumage goes. And the colors, so spectrophotometry out there in the jungle, trying to keep it dry in the rain, looking at collar and belly colors. And so you can see here, these are dot plots with these confidence intervals, and they all pretty much overlap, except this one down here, which is belly color. So we have that Klein for you to see over here on the right for belly color. Contemporary, it looks like it has moved further towards the white-collared populations, which would be on the right here, than how it used to be historically. So it's moved about 9 kilometers based on this signal, which is really fascinating because we think that this yellow yellow belly versus olive belly. The olive belly has more melanin. These darker feathers could be associated with higher aggression, which in a lek breeding species could be very important for lek placement. So overall, what we found, this hybrid zone appears stable over much of the genome, but we have some evidence that one of these sexually selected or proximally sexually selected traits on males may still be moving over this time period. So our next question was, why is this hybrid zone moving? So we wanted to assess hybrid fitness in the field to find out, which is where a bunch of that field work comes in. So we wanted to see just, is there an apparent difference in the survival between hybrids and the parental species? So we were out there misnetting every day and did mark recapture on 3 focal populations. So 1 candy eye population, 1 vitellinus, and we picked the hybrid population that identified as that hybrid center, that very admixed population 9 as our hybrid focal point. So here you can see the average apparent survival estimate between these 3 populations and the associated 95% confidence intervals. And you can see that it looks kind of like the hybrids maybe are actually trending higher, but these confidence intervals do overlap. They're not significantly different from each other. So overall, it seems that survival rates are about the same, if not hybrids doing actually slightly better. So once they're adults, they seem to be fine. You know, nothing much changing there. So next we wanted to look at the difference in reproductive success. So I had noticed that while I was tracking down these nests, which is really difficult in these forests, a lot of these hybrid nests seem to only have 1 chick hatch. They all lay pretty much 2 eggs, right? Sometimes 1 egg, but usually all the manakins are allowed 2 eggs. So a lot of these hybrid nests had 1 egg not hatching almost consistently. So I took a look at this. There's a precedence for lower hatching rates in other hybrid zones. This is seen in chickadees, and birds will often show from either inbreeding or outbreeding depression, something happening with embryonic development in these eggs. And obviously fewer offspring hatching would directly indicate lower hybrid fitness. So here we have this table of a total number of nests that I could track long enough to confirm if the eggs were viable or not. And we then underneath that have the number of nests where 1 or both, in some cases, of the eggs failed to hatch in that nest. So 70% of hybrid nests had 1 or more eggs that did not hatch versus the parental populations had less than 30% of nests with any of the eggs failing. So there appears to be something going on with hybrids having lower hatching rates and thus lower reproductive success. So what I'm doing next is doing a more deep dive into the genetics behind this. So because there's this reduced offspring viability that we're seeing in this hybrid zone, we think that this might have to do with genetic incompatibilities, right? So now we're looking into the genome to look for these patterns. So assessing for genetic incompatibilities. We found this overall stable hybrid zone with reduced hatching rates in hybrids at the genetic center that we're not seeing in the parents. Our next aim is to find regions of this genome that contain genetic incompatibilities. And hopefully these regions will contain genes or a genetic material associated with embryonic development, which would directly show in reduced hatching rates in this phenotype we found in the field. So we used that fancy new mCandiI genome for this part of the study, and we looked at genetic divergence and linkage disequilibrium across the genomes of these birds to find candidate regions in the genome that might contain incompatibilities. So how do we find genetic incompatibilities in hybrid genomes? So here we have a cartoon example of what we're looking at. So if these are hybrid genomes, we're looking at blue and red parental segments. We have like a red parent and a blue parent in hybrid genomes, and there's 3 main outcomes we can see. So if things are combining neutrally, we would expect genes to recombine randomly and freely, like we see here on the left, with the introgressing parent's DNA getting broken up into smaller pieces as it can freely recombine in these hybrids. Then if a trait is beneficial, such as maybe the yellow color, for example, in this study, you'd have an adaptive region that would approach fixation or become fixed of this beneficial allele in hybrid genomes. If it's beneficial, you expect it to spread. However, what I'm looking for is here in the middle. So if our hybrids are some sort of disadvantage from the way these genomes are recombining, these maladaptive regions would purge these bad segments of the DNA from the introgressing parent, most likely, to make sure that these gene combinations work in hybrids, right? So in these regions, we would expect to have everything be from one parent versus the other in ancestry. We would expect there to be higher genetic divergence from the introgressing parent in this region, and we would expect there to be a linkage disequilibrium block or LD block in this region, showing that it's not freely recombining in hybrids. So here I have some of these hot off the press results as an example, because working on this literally this week. Here we have genetic divergence in this hybrid zone. And I used Phi-ST. So this is just a haplotype measure of relative genetic divergence, but it works the same way as your regular FST. So on the x-axis here, we have the position in the genome. On the top panel here, we are comparing all 9 of the hybrid populations against Vitellinus. In the bottom panel, we're comparing them all against Candii. And there's a lot of lines on this graph, so don't worry about it too much. It's all about the individual lines. I'll go over that in a second. So it's hard to see here, but there's this gray dashed line in the back of both of these panels that is the comparison between the 2 parents. So you'd expect that to be one of the highest, right? They're 2 separate species. Their genetic divergence is going to be high, right? On the top panel, you can see this population is 9.5, and when I added at the bottom, it's pretty much Vitellinus. These are mostly Vitellinus birds, so they're not very different. The main pattern to pay attention to here is that you have population 9, that most admixed population I mentioned, that genetic center, kind of pops out in the middle of both of these panels. Population 9 is more Vitellinus-like than other hybrid populations. It's more distantly related than Candii, but it's kind of coming out in the middle. So it has a different pattern versus every other hybrid population is always matching, right? All the other hybrids are eerily close together up here and at the bottom, right? So just population 9 is coming out as being this different population once again. So looking for regions of this genome with differentiation from the introgressing parent being higher in the hybrids, we did FST bootstrapping to find outlier regions in the genome. So we found 2 of those such peaks here. So these 2 peaks were outlier regions in the hybrid genomes. There's many others, but this is an example on autosome, right? So in this autosome, these 2 peaks were highly differentiated, right? And so we want to look at these peaks and compare that to linkage disequilibrium. So here we have 3 heat maps highlighting areas of increased linkage disequilibrium in the same region of the genome. So this is the same scaffold in the genome, the same autosome that I showed you divergence on just a moment ago. So red means high linkage disequilibrium, blue means low. So you can see the Candii parent here on the left, Vitellinus on the right. See, they're mostly blue, right? There's a little bit of structure, in Vitellinus, but overall, they're mostly blue. They're recombining freely. However, we have our hybrids up here. You can see that there's a lot of other colors going on in here, right? So this hybrid population is much redder and yellower, so LD is raised. This is thus implying that there's lower recombination in this region for some reason. And interestingly enough, when we compare this to where our outlier peaks were, it's right here, right in that block. Yay, that's exactly what we want. So the next thing to look at is which parent is this derived from exactly? So I'm currently mapping out what these genes and alleles are from which parent. So we're expecting this to be from Vitellinus, Candii, excuse me, right here, because it doesn't want to recombine because it's happy with the way its gene makeup is right now. So stay tuned for that in the future. This is still actively being worked on, but this is some pretty exciting results for genomic patterns of incompatibilities in these hybrids. So with that, I would like to acknowledge tons and tons of people have helped work on this project, and it's been really, really amazing. So thank you to everybody, funding sources, Smithsonian, AOS, and the RCN. And we only have like 1 minute, but I will take any questions now. Thanks.
Great job being on time, everybody. Thanks for your talk, Kira. That was great. There is a question here if we want to stay a minute or two over. I was just wondering, on those unhatched eggs, are they if you were to open them up, is there anything visible in there?
So I did candle a bunch of these eggs, and so a lot of them, they were vascularized. You could see all of the red veins showing. So it wasn't just that they were infertile, they had been fertilized. And some of them even had tiny little embryos in there, but they didn't develop further. And then the other egg did hatch into a nestling and things like that. I had actually really wanted to collect these embryos and sequence them, but unfortunately they didn't get on the permits until 2020, and we all know what happened then, so I couldn't collect them. But yeah, so I thought that was really, there was a bunch of other stuff I wanted to do too, like Haldane's rule, and look at this, the ratio and get the genes from them, but I couldn't sequence these unhatched eggs, and since so many of them were not hatching, like, yeah. Let's see. Oh, and there's another question. So does melanins could also increase with warmer or dry conditions? So this is actually a fascinating question. So the climate has been changing in Bocas del Toro. It's gotten a lot drier. Like every single person I talk to in the province is like, it's way drier than it used to be. It rains way less, right? So that actually could be a thing that's happening. The climate change driving some color changes. Yeah, so maybe. Thank you for the question.
I got one last one. The surviving chick, if you can tell if that's male or female, can you do something with how?
The sample size is too small. So weirdly enough, I would expect the surviving chick to be male, right? But most of the ones that I was able to get blood from that survived were actually female, but the sample size is so small that it's really hard to say anything. But yeah, would love to get more nests. We had to put radio trackers on these females to find their nests because it was so difficult to find them. So it was a journey.
All right, you guys. Well, thanks so much, everybody. Thank you to all our speakers and everybody who attended, and the AOS Student Affairs Committee. Thank you, everybody, for sharing, and I hope everybody learned a little bit about manakins today. All right, so I think that's it. Everybody's saying good talks in the chat. And I think we'll probably leave it at that unless the Student Affairs Committee wants to say anything else. So.
No, I think we're good to wrap up. Yeah, thank you so much, everyone, for coming, and thanks for the great talks today.
All right, great. Thanks, guys. Hopefully I see you sooner than later, everybody. All right, see ya.
Thank you, everyone.